Hello everyone, Dr. Jack Rivazordi, and today I'm here to take you through protist disease. So we're continuing this pathway of cellular basis of disease, and today I'm going to take you through protists. Now these aren't a very famous disease. We always know about the bacteria, we certainly know about the viruses now. Some people may even know about the fungal causes of disease, but very few people focus on protist diseases. I don't know if many people on the street would know what a protist is. But a protist is not depicted here. That's a picture of a mosquito, an arthropod, and that mosquito is biting a human being. And what they might be doing is transmitting malaria. Now, malaria is a protist disease, and everyone's heard of malaria. So let's jump into protists and diseases caused by protists. So first up, just to categorize it, just to put everything nice into our boxes, protists are eukaryotes. So they have those complicated cells with nuclei and mitochondria. So they're on the eukaryote side of pathogens. They are a communicable disease. Even though they're not directly spread human to human, they are spread throughout the population. And it's not a non-communicable disease like cancer or dementia. However, this is a lie. This cladogram or evolutionary tree is a lie, and this isn't quite how we would divide up life. Let me explain. Now, to understand the complicated nature of protists and why that is a lie, we need to first understand this word, a clade. What is a clade? Now, a good analogy for what a clade is it's any branch of the tree of life that you can snip off and it contains a group of organisms that all make sense on that branch, right? And it could be at any level within the tree of life. So it could be kingdom, phylum, class, order, genus, subphylum. All of those things are clades. So let's have a look at an example. Here we have a pretty good, this is fairly accurate. We have a tree of life here, also known as a cladogram. Hence where the term clade, cladogram. This is a picture that shows you the clades in there. So let's take our scissors and see where we can chop this tree of life that makes sense. So we could chop them here, and yes, humans are more related to gorillas than they are to dogs, so this makes sense. It's a whole group of animals called hominidae. Then we have the mammals, so we could chop it here, and this would include dogs and cats and dolphins and everything like that, and those would be the mammals. That is a clade. We could chop it here. Now these are the amniotes, so this includes lizards and birds and dogs and mammals, so it includes a huge number of typically land-dwelling vertebrates. Some of them have now gone back into the ocean like dolphins, but amniotes there. Now, let's ask this question. Are fish a clade? So is there anywhere we could snip on this tree of life that all the tips of the trees would contain, tips of the branches would contain fish? And the answer is no, right? So fish are not a clade. Fish are a made up category that loosely just means animals that swim in the ocean. Maybe they have gills, even though starfish, are they fish? They're completely different, aren't they? So fish is, from an evolutionary perspective, is not an informative term. And the reason for that is there was an early divergence between cartilaginous fish that are mostly made of cartilage and bony fish that are mostly made of bones. Now, some of the bony fish ended up going on land and creating the amniotes and creating the lizards and the dogs and the humans and the great apes, and so those animals are more closely related to the bony fish than the bony fish is to the cartilaginous fish. So I am more closely related to this clownfish, the Finding Nemo fish we've got up here, than I am to a great white shark. And it's quite interesting, right? Because people go, dolphins aren't fish, right? Dolphins are mammals. But that dolphin is more closely related to a snapper than it is to a shark, right? So it almost doesn't make sense to group the term fish from an evolutionary perspective. So they're not a clade. So why am I talking about fish? Well, let's do the snipping idea. Let's do the snipping idea. Can we cut the tree of life to cut off a branch that contains protists? Let's see if we can do that. So here we have a tree of life, And this tree of life contains a lot of single-celled organisms, right? So there's a huge number of single-celled organisms, some slightly collaborative organisms, but still mostly single cells. But then over here we have a branch of the Merozoite. Now Merozoa is another name for animals. So here we've got fish and worms and arthropods, but also humans would be on this branch right here. Over here we've got fungi, so that's mushrooms and yeast. And over here, it's not depicted on this graph, but over here, coming off this green branch over here, would be land plants like trees and everything like that, right? Now let's label the protists on this tree of life. Everything with a tick is a protist, right? So virtually everything here is a protist. Now, the problem with this is we've got fungi here. So some protists are more closely related to fungi than they are to other protists. Some protists, this protist here, would be more closely related to humans than it would be to a protist over on this side of the tree right here. Now land plants come over here, so there are some protists that are more closely related to land plants than they are to other protists. So what that means is protist doesn't really have an evolutionary concept there, right? They didn't, although they all came from a common ancestor, undoubtedly, eukaryotes, they broke off the branch later on once they'd committed to a branch, say, to become fungus, right? So they do not make sense in terms of an evolutionary perspective. They are not a clade. Now, this is supposed to be a medical degree, so why do we care about evolution and clades? Now, I've said this before and I'll say it again, you cannot understand biology without understanding evolution, and evolution applies to all of biology. Even being a doctor, it would be important to understand evolution from something as practical as being a doctor. And I'll go into that right after I give you the definition of protist then. So what is a protist? Well, it's a real bucket term. So protists are any eukaryote organism that is not an animal, plant, or fungus. So animal, plants, and fungus all have really quite tight definitions. We define them as that, and they are clades. We can chop them off, and they would all be on one branch. But protists are kind of everywhere in the tree of life, and so we kind of just dump all these sort of dead-end solo organisms, often single cellular, into the term protist. A lot like fish, right? They're not evolutionarily related, but they kind of look similar, so we just lump them together like fish. Now, this is super important from a medical perspective, and I'll tell you why. So azoles are an antifungal drug, and I'm going to go in my next video actually about what azoles are. So azoles are an antifungal drug, and they work against most fungal infections in the clinic, and that is because fungi all have a few things in common. And so if you have something that's unique to fungus, you can target it with a drug to get selective toxicity. So it won't kill us because it's targeting something unique to fungus. Antibacterials, we have broad spectrum antibiotics like amoxicillin that are effective against most bacteria, and that's because they target something unique to bacteria, and because they're all evolutionarily close together and they're a clade and we can snip them off as a single branch, they have those molecular things that are uniquely in common with all bacteria. Same with fungus, right? Because they're a clade, they're all genetically descended from each other and all related to each other, and so they have these unique molecular things that identify them as a unique thing. Fungus. Protists are everywhere. So there are no drugs which are effective for all protist infections. Each drug is fairly specific to a very, very small group of related protist species. And that's because you couldn't find something molecularly in common with all these protists because they are so disparately related on the evolutionary tree. For example, there are protists more closely related to me than other protists, so if you tried to design a drug that targeted all protists, you would probably get me in that list too, because how could you molecularly distinguish me from a more closely related protist than a protist that's distantly related to both of us, right? Okay, so that's why it's important. That's why it's important that we understand these all from an evolutionary perspective, and biology. Evolution is the foundation of all biology. All right, so let's do a quick, just little snapshot about causes of death. So E. coli, it causes around 250,000 deaths a year, mostly through diarrhea. That's a bacteria. Influenza, this is a virus. It causes deaths around 500,000, a little bit more per year, depending on the season. Staphylococcus infections, a little bit more. It gets up to 600, 700,000. It's not important that you know the exact numbers, but it's important to know magnitude. So malaria kills around 450,000. So this small group of protists are involved in killing a huge number, and they are uncomparable to these very famous, well-known diseases. We need to be thinking about protists. And when we're talking about cellular basis of disease, you cannot exclude malaria because it is right up there with influenza and staphylococcus and E. coli as a #1 killer. Another little important note is often the people who die of malaria are the young. And so in terms of a cost to human life, malaria is massive, right? It's huge. Right, so let's go through the life cycle of malaria. Now, there is a bit of jargon here, so I'm sorry about this. But trust me when I say I have stripped out about 60% of the jargon in this life cycle. So this is just some important jargon to bring you up to speed on the rough life cycle of it. So, a mosquito that is infected with malaria will bite you. And when it bites you, it drains your blood. But it doesn't want that blood to clot. So what it does is it secretes saliva into the bite wound. And in that saliva is anticoagulants, something that stops the blood from clotting. But also in that saliva can be the sporozoites of malaria. Now, sporozoites, it's a funny term, right? This is how I remember it. So it's called sporo because it's the infectious agent, right? So spores we often associate with infectious agents. There are numerous things that have spores. Fungus have spores, but even some bacteria have spore-like things that we call spores. And they're really just an infectious agent. So that's why they're called sporozoites. Zoo meaning animal, and ites, not sure where that comes from. I think it might mean a group. So, you know, if you're from Hobart, you might be a Hobartite. And so this is a group of sporozoites, right? So sporozoites, now these dudes are awesome, right? Like, I know they're horrible. They're a disease, but they're pretty cool. Here we have one here, and you can see that they're very mobile, and they swim round and round in circles. They're quite large compared to the Merozoites. And what they're doing is they're very mobile, and they're that shape so that they can shoot their way through the tissue to find a blood vessel. They're looking for a blood vessel. They'll jump into the blood vessel, and then they will flow around the bloodstream, hunting out the liver. They have to get to the liver. This is the liver here. Now, in the liver are liver cells called hepatocytes. So hepatocytes are liver cells, and they will climb into the liver cell, and then they will, inside the liver cell, they will begin to divide. And this is mitosis, it's asexual division. They will divide to form a different structure, a different morphology than the sporozoites. They form these much more circular, tightly packed, smaller versions of malaria called Merozoites. Now, I have a terrible way to remember this. So the word more is spelled M-O-R-E. So when the sporozoites become, there are more of them, they're more zoites. But then you swap the O and the E and you're at Merozoites. I really don't know if that helps, but that's how I remember it. It's like that there are more malaria cells. So there are Merozoites. Merozoites. So here are Merozoites, and they hunt out blood cells. They go into the blood cells, and then they actually consume the blood cells from the inside out whilst dividing and dividing and dividing in asexual reproduction, so mitosis. But they can also burst out of a red blood cell. They eventually kill it, and it bursts out, and they can infect more red blood cells. So you can see the disease aspect of this. They are destroying your liver by going into your liver cells, intracellularly dividing, consuming your liver cells. They're bursting out to your red blood cells. They're going into your red blood cells, dividing in there, bursting out of that. But there are actually more disease mechanisms that I'm about to go into. But they can also produce gametocytes.
Now, cytes just means cells. We're going to see that a lot. And gametes just means reproductive cells. So gametes, like a male human gamete is a sperm. So gametocytes makes perfect sense. It's the first thing that's named half decently in this life cycle. The rest of them are named terribly in my mind. But gametocytes are pretty good. So they're now floating around the blood, these gametocytes. And then an uninfected uninfected mosquito will come in and suck up the blood containing the gametocytes. Now these gametes will then fuse to form a zygote, and that's what happens in all animals. When one gamete meets another gamete and fuse, we get a zygote. That zygote then turns into a sporocyte and tunnels its way from the stomach of the mosquito to the salivary gland. So it will then form this worm-like sporocyte and then tunnel its way to the salivary gland to then be injected into the next human being through the salivary gland. Very cool pathway. Now, here we have a red blood cell, and this actually hasn't been stained, and you can actually see the malaria, and that's because malaria create these crystals that are pigmented called hemozoin, and I'm going to go on to explain that a little bit later. Now, the spleen. It's one of the weird organs that you often don't learn what it does until much later. We're going to go into that in detail in one of my later immunological videos because it's a really important immune organ. But one of the major roles of the spleen is to filter your blood, in particular your blood cells. So it filters out dysfunctional and dying blood cells. Your blood cells don't last forever, so you need to take out the dysfunctional blood cells and they will be replaced from the bone marrow producing blood cells. So your spleen is always filtering your blood, and they will filter out any blood cell that's infected with the malaria, the Merozoites, right? So the Merozoites don't want to go to the spleen. They don't want to go there because they're going to get eaten and consumed by the immune cells inside the spleen after they've been filtered. So what they do is they force the production of these sticky proteins on the surface of the red blood cell to allow them to adhere to the blood vessel wall. And that's because if they adhere to the blood vessel wall, they aren't going to the spleen. So here we have a beautiful histological image here. You can see the malaria-infected red blood cell here. You can see that pigment that I was talking about, that hemozoin. You can see that pigment there. And then this is a capillary. You can't really see the walls of a capillary. But these are red blood cells. 1, 2, 3, 4 red blood cells are stacked up behind that capillary because we have a roadblock. The infected red blood cell has blocked the path of the capillary. Here's a diagram here where we can see it. The infected malarial cell has blocked it, and now red blood cells are stacking up behind it. Now you can imagine that's not good for the host to get these kinds of clots all over the body, and it can actually have larger blood vessels. So these sticky red blood cells that have the malaria merozoites on the inside will just stick to the wall of a larger blood vessel, not a capillary, maybe an arteriole, which is a small artery. And then we can, once 1 or 2 stick, it slows down the blood flow, allowing more to stick, which then will cause an eventual full blockage of the larger blood vessel, and it will cause a much larger clot than in a capillary. It's much more damaging to block a larger blood vessel than a smaller blood vessel, but malaria can cause the blockage of both of those. Now, this can cause large clots. This is a blood vessel here, and we can see it's absolutely packed to the gills with cells, right? So this is a clot right here. So that's one of the results of the malaria parasite, these clots all over the body. But it can also damage the blood vessel wall, and any kind of clot can cause a pressure and a swelling behind that clot. And so called an aneurysm, and that can burst, and so that can cause hemorrhage. So the malaria parasite causes both clotting and hemorrhage, or the leakage of the blood vessels. You can see all the red blood cells are just spread throughout. Now this tissue, unfortunately, is the brain, and the brain is an incredibly sensitive organ for this kind of thing to go on. Now these images, I have to give a shout out because these are really good friends of mine. Have all come from this research paper by Patrick Strangwood. We play Age of Empires 2, great game, highly recommend it all the time. Michael Haley, he's my jiu-jitsu partner. Tova Shaw, she's just an amazing person. And yeah, so this is actually like 3 of my best friends published this paper. Fantastic research. Anyway, have to shout them out and name drop. They're awesome scientists. Anyway, okay. So, What are they doing in those red blood cells? Well, Merozoites consume hemoglobin, and hemoglobin is the protein complex. There's more than one protein. It's a protein complex that transports oxygen around our blood. So we need that. It's really not good if they are eating that. So they eat this hemoglobin, and the hemoglobin contains iron. Iron is actually an essential iron in there. ION. It's an essential metal ion in there for oxygen binding. So we need the iron in there for the oxygen binding. That's why being iron deficient causes anemia, low red blood cells, because we need it for this hemoglobin. And the Merozoites eat this through a process called phagocytosis. It's where they crawl around it and suck it in, basically. And when their membrane crawls around the hemoglobin and sucks it in, that forms what's called a phagosome. Now, a phagosome is this internal vesicle now, basically, inside the cell, and the phagosome will then fuse with a lysosome. Now, a lysosome is a specialized organelle. It's basically a vesicle that's been acidified, so now it has a very low pH, a lot like your stomach, and it's full of digestive enzymes. And so the lysosome will then fuse with the phagosome to form a phagolysosome. This is kind of good naming. I quite like this. This is quite good. So a lysosome will fuse with that, acidifying it and putting digestive enzymes in it. So the digestive enzymes will digest down the proteins and the nutrients inside that, and that will then be released and utilized by the cell. But the iron is a very dangerous metal ion. It catalyzes the production of free radicals, which are very damaging molecules. So high concentrations of iron is very damaging. It's kind of a toxic molecule. So how the malaria parasite deals with this is it crystallizes it. Once the iron is in a crystal, it can't do any of those toxic reactions that it can be involved in. And so it's nice and safe. So the malaria parasite actually creates a vacuole full of this toxic crystal called hemozoin. Now, it's a brown crystal, and that's why we can see it in all those other images. So it's also called malaria pigment sometimes, because it's almost like a self-pigment of the merozoite, is this hemozoin. It's a pigment, this hemozoin. So, oh boy, Donald Trump got all excited by this anti-malarial drug called chloroquine, for the treatment of SARS-CoV-2. It doesn't treat SARS-CoV-2. It has negative side effects. In fact, a few people died because they went out and got chloroquine and took it when they shouldn't have. So anyway, only take chloroquine in if you've been prescribed by a doctor, and they'll probably be prescribing it because you have malaria. So how does it work? It's an anti-malarial drug. Well, what they do, now, you'd be surprised how sure we are of the mechanism of action of a lot of drugs. We prescribe a lot of drugs without really 100% knowing the mechanism of action. Paracetamol was probably only identified the mechanism of action. It's still debated in 2017 with a fantastic paper. It involves cannabinoids, by the way. Anyway, off topic. Anyway, so this is how we think chloroquine works. Hydroxychloroquine works. And what we think it does is it neutralizes lysosomes. So that prevents the lysosome from acidifying the phagosome. And so you can see here it's got a neutral pH, right? The chloroquine goes in and neutralizes the acidic environment inside the lysosome. This prevents the crystallization of the iron compounds, and then the iron compounds do those dangerous chemical reactions to cause the lysis and death of the cell. So harmful uncrystallized iron ends up killing the cells. So now you can kind of see, I started the video with this, you can kind of see that that is not a drug that's going to work for all protists, right? It's only really going to work for malaria. And that's what I was talking about, right? Understanding that protists are so evolutionary distant that we won't be able to develop drugs for all of them. We'll have to target them one at a time. Now, I'd love to go into more examples. There's a brain-eating amoeba that's also a protist that you can get from hot springs. But I don't want to flood you guys with too much information. I just wanted to give you one good example of a protist disease and how we fight it. Next up is fungus. Now, fungus are another eukaryotic pathogen, so I'm really excited about this.